Alright, so on we go. This is the third of five talks on human physiology's immune response. So far we've covered the innate response in the first one, and now we've looked at the basics of what makes up the specific immune response. What we've learnt from that is that we have really two sides of the specific immune response. We have the cell-mediated side run by T cells, which is what we're going to look at now, and then we also have the humoral-based side, which is mediated by B cells, which will be in the next lecture. Alright, so let's not waste any time, let's just get on with this. So we've done this one on the specific side, so now we're going to look at cell-mediated specific immune response. So cell-mediated is done by T cells. This is all about T cells. So let's get that up there. T cells. So there are four types of cell-mediated T cells: cytotoxic, helper, suppressor, and memory. So those are our four main types of T cells. Now, as I said in an earlier talk, a T cell comes from the thymus, and it's part of the endocrine system. It sits up here under our sternum and is active for about the first 30 years of our life, and after that it sort of shrinks down and does much less. T cells after that point tend to be set up in the lymphoid tissue, such as the lymph nodes, but originally it's the thymus that creates these cells. So what do we have? We've got these four. So cytotoxic, well, cyto means cell, toxic means toxic, so deadly to cells. So this is the cell killer. It's also known in papers, you'll see it written as CD8 cells. So the cytotoxic T cell, or CD8, is the killer cell. It has the ability to latch onto the major histocompatibility complex class 1 proteins, and you'll find them on every host cell that has a nucleus. Pretty much every host cell. I mean, for our intents and purposes, just think of it as all cells that have a nucleated cell, because that's good enough for our thinking right now. So what does this do? Well, each cell with a nucleus in your body basically has one of these proteins on its surface, on its outside, like a jacket, if you like, for you or I. And it basically has proteins built into it which come from inside the cell, which show that they're the same proteins that are inside the T cell or inside any other cell. So it's basically saying, hey guys, I'm one of you. And as I said earlier, that's if you happen to be into football in Michigan, you're probably going to be either a Spartan or a Wolverine. There are other options, of course, but those are the two main ones. This is from a guy who's Scottish and knows absolutely nothing about it. Anyway, so if you're a Wolverine, you went to MU, I think I'm getting that right, then all your friends, fans, etc., the same thing, will all be wearing the blue shirts. Whereas if you're a Spartan, you'll be wearing the green and white shirts. But this is basically what your cytotoxic or CD8 cell is looking for. It binds onto a host cell, well, one of your cells, and looks for the normal proteins which it has itself. If it finds that, we're all good here. Well done, you're a Wolverine fan too, enjoy the next game, or Spartan if you happen to be a Spartan, and it moves on. So that's the focus of the cytotoxic. If, however, it binds onto an MHC class 1 and it doesn't find proteins based from inside the cell, which are normal proteins, such as in a cancer cell, which is, you know, one of your cells which is now growing abnormally, then it destroys it. Now, how does it destroy something? Well, it has a number of ways it can do this. Perforin, which is those little proteins that we talked through in the first talk with natural killer cells. Natural killer cells, or NK cells, and CD8 cells have a very similar mechanism in which to destroy, the target cell. And these perforin proteins, I'm not going to try and do that thing again, but they basically create a hole. They're individual proteins and they all go together, making a hole in the center. And of course then all the stuff from the inside of the cell comes out and the cell dies. So they put perforins into the cell wall, but they don't just stop there. CD8 cells, or cytotoxic T cells, also produce lymphotoxins. Now these toxins are produced by lymph cells, CD8 cells, and they destroy the DNA of the target cell as well. Now, why do we have to do that once we've already killed the cell? Because that cell, if it's been compromised by a virus, for example, may now have altered DNA, and that DNA could get into other cells and incorporate itself and carry on being a problem. So this is like a secondary stage destruction to make sure everything's been destroyed, it's not coming back. So kill the cell by just putting a hole in the wall, kill the DNA of the cell to stop any potentially abnormal DNA getting out, and it also has a tumor necrosis factor, and this basically is aimed at cancer cells. And tumor necrosis factor destroys cancer cells over a 2 to 3 day process. So the CD8 cell, or cytotoxic T cell, is the killer of cells. It does this by using the MHC class 1, which is on the actual cell it's investigating, and this usually will have self proteins there saying, I'm one of you guys. But to use that Wolverine Spartan analogy, if you're a Wolverine and there's Spartan cells in there, well then they will be taken out. Or if you're a Spartan and Wolverine cells are in there, then they will be considered not host, they will be removed as well. Okay, so that's our cytotoxic T cells talked about. So next one, helper T cells. These are also known as CD4s. Now, we came across these in the last talk because we were talking about antigen presenting cells. And the helper T cell does exactly what the name suggests. It helps coordinate the whole of the immune response, including the innate and the acquired. So innate being the non-specific and acquired being the specific. So here on this side, this is pretty much the same diagram as you have in the PowerPoint lectures, so I urge you to look and listen to that as well. But here's your macrophage, or your B cell for example, and it's engulfed a bacterium. Let's just get the right colour here. So the bacterium has been essentially destroyed. Inside it using lysozymes. There's the major histocompatibility complex class 2, and that's sitting on the surface. So this macrophage has now become an antigen presenting cell because it's presenting the antigen from the bacteria on its MHC2. The helper T cell, CD4, is able to bind onto this and then look at what this is. That's important because it can then, once it's bound onto this and realize that this is, an invader and needs to be taken care of, it can do 3 things simultaneously. First of all, it can stimulate macrophages through a macrophage activation factor, and that chemical will cause the macrophages to become more active. Macrophages are part of the innate system or non-specific system which engulf bacteria and viruses and all the rest of it. So they become more active. It also triggers inflammation response. So again, why do we have an inflammatory response? Well, simply to get more of the white cells present at the site of the trauma or the infection. So you're increasing blood flow to that area to get more white cells there. You're keeping the blood thin so they don't, because all the cells don't basically clump up together and cause clots or things to form, or agglutination processes, and they become leaky, so the cells can then leave and get out and attack. Now that's something that when we look at the inflammatory response, I do urge you to look at the PowerPoint, because you'll see the process of diapedesis, and I'll write this down for you. which is the process of a white cell. This is a blood vessel with like capillaries there. The white cell can push out and leave the blood and enter the tissues. So diapedesis. So this is part of the inflammatory process, which is stimulated when a helper T or CD4 latches onto an antigen presenting cell. So the infection's taken place, the neutrophils have got there, they've started killing off the bacteria. 12 hours later the macrophages turn up, they start producing antigen-presenting cells. At this point the CD4 cells latch onto them, continue and increase the macrophage response, increase the inflammatory response to keep getting fluid, keep getting more and more white cells there, and also increases the chemotaxis of the non-specific cells. And this is the diapedesis, where the cells are leaving the blood and going toward the damaged tissue, because there's a chemical breadcrumb trail from the damaged tissue entering into the blood to help them. So that's one of the things that the helper T cell does. Secondly, it actually stimulates the humoral immunity, and this is through a chemical called interleukin-2. Leuc is white, so inter between. This is a chemical sent between white cells. So this particular one was the second one found, so that's why it's called interleukin-2. It's released by a CD4 and arrives at a B cell. So it's one white cell's talking to another. So between white cells. And this causes a clonal selection of the B cells to produce antibodies, because we now know what the antigen determining factor is, because the helper T has bound to it. There is the antigen determining factor. The antibodies, which we're going to look at in the next one, that have to attach to that are going to have to, because antibodies are Y-shaped structures, are going to have to be able to attach onto that in such a way. So that interleukin-2 is sent with the information to select for the B cells that produce the correct antibody that will attach onto the antigen and cause it to agglutinate or clump together. And finally, an interleukin-1, this was the first of the chemicals that were discovered between white cells, goes from the antigen presenting cell via macrophage or a B cell, etc., and that goes to the CD8 cells. And again, that tells them what to look for in the MH class 1 cell. So you'll see this abnormal protein, guess what, this is what it's going to look like. So the non-specific system is stimulated, and both sides of the specific immune system are stimulated by this antigen presenting cell helper T cell complex that's formed. So it's an incredibly powerful part of the immune system. And basically, to very quickly mention a disease which has taken many lives over the years, HIV. So the HIV virus lives in the CD4 cells and destroys the CD4 cell. So what you know from HIV, which becomes AIDS, or can become AIDS, is it takes that cell class out. Now that by itself should be able to tell you just how important the role of the helper T cell or CD4 cell is in organizing the entirety of the immune system to respond. Because people without helper T cells, because of an infection like HIV becoming AIDS, basically are open to all types of infection and unable to defend themselves against it. And of course the result of that is death. So we've got two more, which I'm going to briefly mention because we focus on these two at this level. Suppressor T cells. Now you think, well, why do we need a suppressor T cell? Don't we want them to get the job done as fast as possible? We absolutely do. However, when you're basically making a clonal selection of all these T cells because of this stimulation here, so they're all out there picking up on that particular antigen determinant and they've all been selected. Now clonal selection is like taking the right copy, if you like. This is the one, this is the sheet I want to, put up on a poster or something like that. It's all spelt right and it looks good. And then you make loads and loads of photocopies so you can go around the town putting your poster up. Well, this is the same as T cells being clonally selected. You found the one that will bind onto the antigen determinant. So that's the one that will be able to destroy cells which have been infected by this particular agent. So you clonally select them, so now you've got loads and loads and loads of the same, basically photocopied, and they're out there to do their job. The problem is if you produce too many of them, they'll start getting a little overactive and start taking out things they shouldn't. They'll start looking at things saying, yeah, you look a bit suspicious to me, I'll take you out too. So suppressor T cells basically reduce the activity of the cytotoxic Ts or CD8s once the infection is taken care of. They also act to resist autoimmunity. Now, autoimmunity is something that we look at a little bit in the PowerPoint, but autoimmunity is when your immune system starts attacking your host cells. So this whole MHC class 1 host cell thing is not working correctly. The antibodies, for example, of the B cells or T cells are attacking cells that they shouldn't. This is becoming more of a problem in our society. Our levels of autoimmune disease have exploded over the last 40 years. And the reasons for that are multitude, but at the same time, it's very difficult to pick out and say, well, if we make these changes, this will go back, because we're fairly far along the road now of these changes having taken place in our environment. And lastly then, memory. Now, we call this the specific. So it's got specificity through MHC class 1 and 2, but also memory is important. Remember right at the back, right at the start of the last lecture, we talked about specificity and memory. Now memory is important because once you've clonally selected these cytotoxic T cells, so as soon as they recognize that antigen, bang, they're on it, they destroy the cell that has it. Well, the infection's taken place, it's gone, it's finished, the suppressor Ts have basically got rid of all of those cytotoxic Ts now and everything's gone back to normal, and then someone else in your house gets the infection because it's been passed on, and then suddenly you're re-exposed to that infection, and so the memory cell is still there and is still circulating, so it latches on as soon as the infected agent is back in the system. So it's an incredibly fast response, and it's so fast that it takes care of the invader before you're even aware that anything is happening. So your immune system's taking care of the business and you never become symptomatic to it. So you're going along fine, even though you are still being exposed to it. So these are the four cells of the cell-mediated specific immune response. They are the T cells, because they're made in the thymus, and they are cytotoxic, the ones that kill the infected host cells. Helper T cells, these are the ones that coordinate between non-specific and specific responses of both sides, humoral and cell-mediated. Suppressors, which stop cytotoxic T cells doing untold damage. And memory, so that if you're ever exposed to the same infection again, you don't become symptomatic for it, you just take care of it. So that's the end of this one. So when we come back, we're now going to look at the humoral side of specific immunity. Thank you.